Okay, so welcome back. Let's talk about this Pokemon Pikachu thing. Okay. So as we learned from Shirley's lecture, that if we have this High-SIN map, this contact map, we might be able to first to identify some obvious regions that are interacting much more frequently than each other. So like this triangle and this triangle. Okay, so we call these regions TAT. Right, this is the regions. We can consider that to be the independent interacting unit here. But we are more interested in the thing that we are more interested in would be the unexpected interaction, the unexpected peaks in the blank region, in the blank area. For example, here we see we can tell directly, even as a human being, that there is some interesting peak happening here. For this one, we are not that sure. If we draw another box here, we are pretty confident there is no such thing. So this Pikachu is trying to say, now I want to train a computer vision model, image-based model, where we take each input like this, diamond region. We take the input like that, and then we're trying to make a machine learning model to, to classify whether this is supposed to be called as a positive peak or it's just a random noise. Okay, so that is the idea. So for this homework, we only need to use some of the pre-trained Pikachu models. And just give you a very brief introduction of this Pikachu, the methodology. It is taking, like I said, taking the input grids, taking the patches, and we manually provide them some labels, whether it's negative or that's a positive. So the positive labels are the things we directly get from input. So we know already which part can be positively interacting. For the negative samples, we just randomly sample some of the regions on the huge map, because the reason we do not specify where do we want is because we want to cover most of the features on the other part of the graph, and also we want to try to make the negative samples more diverse, but also try to avoid touching any false negatives. So this way, we basically just define positive cases, and then we random sample a few negative cases, and then you can train a binary classifier. This classifier can be neural network based, can be random forest. I believe this time we are using a random forest classifier. And you can also use this as VM, a lot of other things. Doesn't matter. It's just a question we are trying to solve, trying to answer here, is to take a patch and trying to assign each patch a label, whether that is a peak or not a peak. And during training, you would still need to do cross-validation and parameter tuning, like we talked about in the first homework, or at least the first session, about how to do well machine learning. Okay, so that's the methodology, the concepts. And in terms of how to run how to run Pikachu, is that after you load this environment, there will be a command called Pikachu becomes available in your from your terminal directly. So now you want to say, I want to use Pikachu to score for a chromosome, a specific chromosome, or I want to score for a specific genome. So it has these two options. Here we are more interested in this, specifically interested in this 21st chromosome. So we would use this score chromosome and try different resolution, different ways of the binning, different whether do we use balance or not, what are the CRU data we are using, and what is the model we are using. So this CTCF model is a pre-trained classifier that we loaded from another kind of repository. So we also provided you this model in the P3 data. Try looking into that and trying to load this model directly for Pikachu. So this line would use this model, use this classifier to give a score for each of the region, for each of the bin region, a score for each chromosome or for each genome, to provide them a score for whether this is likely to be a positive or negative. And then we can now go into this output folder, and then you do this Pikachu again, because now you would have a very high resolution thing, and you do not want that high resolution, so you can do a pooling from this chromosome, from this first stage outcome to get the ultimate outcome we want. Okay, so we will save it as a bad P. I would call it bad P file. So as you can see, for all these hyperparameters are something we can tune during model generation and also during model application. So here for the homework, we can just set the resolution to be somewhere between 1,000 to 10,000 would be enough for both visualization and also for classification. Okay, so that gives you the peaks and the file of the peaks. Now we also ask you to do some visualization of the results, and to do that, HiGlass is one very powerful tool to directly use it. So I know you have already run some Python thing, but I'm not sure whether you have used Jupyter Notebook. So for this HiGlass, first of all, it has to be an interactive session. So it would require you to use Jupyter Notebook. And because you want to do this interactively, it is a little bit hard to do this with Canon. It's not impossible. You need to do a few port projections so that you can directly see the interactive kind of UI website that you can put in the Python code as just like what you put in your RStudio. So for this question, I would highly recommend you to download the result files. Make sure you only download the required files. For example, here we only need this I'm cool file, which is what we generated from part 2.1, I think. The multi-resolution cool file. We only need this file, and to run everything here in the local Python. The reason of that is, as I said, so that the Jupyter Notebook can be easily attached, and you can directly see your output. If you have any question regarding how to set up the Python environment, how to install this Jupyter, try to go back to in the first session on how are we supposed to use Python. But for now, you can just try to start from install this Jupyter together with this HiGlass in Python. If you see this installation successfully finished, you are good to go. You are good to calculate directly to open a Jupyter Notebook and then to load your HiGlass over there. If not, go back to the previous slides, to previous labs. Okay. So now after you install this Jupyter, you need these 2 additional lines to link the HiGlass together with Jupyter. For now you don't have to know what these 2 lines are doing. It's just linking the HiGlass graphic interaction, interactive UI with Jupyter. And now after you successfully set up everything, you can just type in Jupyter Notebook. Let me show you what that means. Okay. I am showing you the entire window, right? I hope I am. Well, let me double check because I don't have an audience now. New share, just to make sure I'm sharing you the entire screen. Okay, hopefully nothing changed, but just in case. So in order to do that, we need to let me close everything here. Okay. So as you can see now, I'm at my home folder. Okay, it's not the Canon anymore. It's my personal laptop. And the thing we are going to do is to, create a kind of environment that we have already created before, right? But now this time we want to activate some of the previously installed Python 3 environment. For example, here I'm going to use this environment. I just rename it as Conda 3.8. Because here I would have a Python 3.8. So this is just my way to keep track of environments. But now I'm in this environment. Now I can directly go into Python, right? I can import whatever I need, right? I can import, for example, HiGlass. That should also work. I can do actually everything I want to do, but I just would not be able to see the graph, the figures, because of the nature of this terminal interaction UI. Okay, so in order to have all this graphic interaction, we would need to call this Jupyter. So we just give you a sense of how do we install it. So you just copy paste these 3 lines here. Okay, you install it. As you can see, it's all satisfied, and because I already installed it, but when you do it, it would take some time because you need to, as you can see, to load all of them to your local machine. It wouldn't be a very large space size, but it would take some time to install. And to run these 2 lines, to link Jupyter with HiGlass, you will see an OK if the linking is successful. Now, if you have everything settled down yet already, you'll be able to call Jupyter notebook, and it would automatically generate a server. It automatically generates a server and give you saying now this Jupyter kernel is running behind in the backend, and now you can access this Jupyter by using, for example, this link, or it's actually automatically generate a link for you. So when you try it, you will be able to see. But for now, let me just copy this line with the link together with the token. Okay, so just imagine I don't have it. Right, I can just copy paste, and now I have this Jupyter. Okay, and now you want to navigate to your data folder, to your result folder, and create a new Python 3 notebook. I already created one. Yeah, the connection is failed because the original kernel was killed. I was trying to demonstrate this. Okay, so now the first thing we want to do is we want to import all the functions we need from HiGlass. Okay, HiGlass is a very nicely maintained package. The only function you need are the HiGlass itself. So you just you just load this HiGlass information, and now the second step is to load the data set. Oh, sorry, it's not connected, so I probably need to restart the kernel. Okay, so now let me load the HiGlass. So this is loading the environment, and now I'm loading the data set. Okay, so the HiGlass called this a track set, because if you go to the website, as we will do in a second, they call each data set to be a track set. They call each type of figure a tracker. Okay, it's not a very catchy naming, but that's how they do it. So now you have this TS1, which is looking at this cool file. And now you will define your own tracker. Here we are going to track a heat map of that set. Oh, sorry, not track set, the tile set. I should have probably changed tile set. So that is your data input as a TS1, and you track it. And now you want to view the plots, and you want to display it in a server. So you can just use all the functions built in, from HiGlass, and you just run it. So now it's already ready. Okay. And now you want to just call this display, and it gives you a interactive window with hopefully a heat map. Exactly. See. And now the fancy part of this is that you can directly drag this window. It can change to whatever shape you want. You have some buttons here that some of them do not work because they borrow this UI from their web portal. So some of the things you would not be able to use, but some of them they are. For example, you can add a figure at the top of this heat map or at the bottom of this heat map. And for this heat map, you can also change. You can also change. For example, the color. I forgot where the color is here. So you can change the color to different settings. You can generate a figure as you wish. So for the homework, this is what we need. But now let's go to the HiGlass portal just to see what they are providing us. Okay. So this is HiGlass, and we can look at some of the examples. For example, the example could be this one. Let's say, just pick a random one. So HiGlass would allow you to generate multiple figures. For example, here it's probably using different resolutions. As you can see on the right, you have a higher granularity, lower granularity, and on the right, on the left, you have a kind of a lower resolution. And then you will be able to change, the color for each of this. Let me try to do one. Hopefully it would work. It's not working. Maybe this background color doesn't mean what I suppose is meaning. Let's try to change a color code. This is something we can change. Okay, so this portal is providing you the way to generate multiple figures. So for example, here we are looking at 4 heat maps, but we can actually change. We can remove this heat map, I think. Close out series. Yeah. So now we have this tile again. So this is a data set. So this 4 might be 4 different data sets, but for this particular data set, we can now create a new, a new figure by looking at all these publicly available data sets. I just randomly pick one, and then we would have the heat map for that one. So we can generate, whatever we need on this portal. It's very fast. It's using the server to give you the calculation. The only shortcoming is that they do not support all the customized data set. It only has a given set of given collection of data sets that you can play around with. Okay. This time we are doing our own example, so that's why we have to do this locally. But just want to give you a sense of why you have these special buttons everywhere instead of just a heat map. So for this one, for example, originally, sorry, I already closed it. For example, in this one, let me just show you. I can actually, download the file. I can just export the files as a PNG or as a, a vector graph. This would work on the website, but for our own Jupyter, it might be harder. I haven't been able to do that. So you can please feel free to check it out. If you find out how to do it, let us know, let the class know how to do it. Because if you just download it here, it will just download you a empty figure. I really do not know why. So just for this homework, you can just make a screenshot of the heat map. That's all we want to see, so that we know that you learn how to use HiGlass to visualize a cool file or cool file, and you also understand what a cool file is. Okay, so that is part 2.2. Yes. So now let's move on to the next question, part 2.3. The point 3 and point 4 are very similar to each other. Not very similar, but are using the same data, which is the bad P data. Okay, bad P data. So we are again going to use this bad tool, because this time we are trying to find the interactions between between 2 different files. For this question 2.3, now we want to see those ATAC peaks, and we want to see the interaction of these ATAC peaks with the loops we identified from Pikachu. So this Pikachu is giving you the peaks, and it's also giving you the x and y of the peak, right? That's how you define a location in this two-dimensional matrix. So this x and y is called anchor 1 and anchor 2. So here in this anchor location file, this is the results we generated, right? Remember, we would have the interaction in a extended graph version, where you have x and you have y, and then you have the interaction score that is calculated by Pikachu. So now we want to say, for these regions, and how many of these events would overlap with the peaks we identified using other techniques, using the ATAC sequencing techniques. Okay, so ideally we would see some association, but let's try try it on our own. So again, even though now this time we are not looking at the gene expression levels, we are looking at a score that we develop from other softwares. We are still trying to look at intersections between different genomic regions. So again, we have a reference file for a value for different region, and we have another file saying these are the regions of interest. So the function, the command, the software we are going to use is bad tools again. So here, what we are going to do is very similarly, the bad tools intersect, but now just try to put the peaks as the first file and trying to put the anchor as the second file. So again, this is now becomes the peak. It now becomes the reference, the background reference, and now the anchor becomes the locations of interest. So you can calculate that, and you would be able to generate your data set. But just one thing to take into consideration. Now this bad P data is not like the bad data that bad tools would like to see, because it has X and have Y. And here we are actually interested in both anchors. Okay, interested in this anchor 1 and also in this anchor 2. So the way we are going to do is to take the first three columns, which would give you all the locations for all the anchor 1. And we would take the other 3 columns here, would gives you all the locations for all the anchor 2. And then we want to concatenate anchor 1 and anchor 2 together. We do not want to separately to analyze them, although we can. But here we are just concatenating them, and then we're trying to calculate directly this anchor, like in general anchor locations, trying to look at the intersections between the anchor locations to the peaks background. Okay, just one quick note on how to pick columns from Bash. We are using this awk command, and using this specific syntax, it would gives you the first 3 columns of the file of interest of your text file. And again, just to recap, this greater symbol means you are writing your output into a file. So if you want to calculate, now this will give you only the first 3 columns, which means the left anchor. If you want to calculate the right anchors, you just pick from the 4th to the 6th columns and save it in another file. And later on, you catch the anchor 1 and anchor 2 together, put the greater symbol. That gives you a concatenated version of all the anchors. And now you have all you need. You have the peak background file, you have the anchor file for the locations, and you are good to run these bad tools. Okay. And now we just apply these bad tools. As I told you, for 2.3 and 2.4, they are very similar to each other. For 2.3, you are using this ATAC peaks as the background. For 2.4, we are actually more familiar. We are going to use a gene expression specifically for chromosome 21 to generate what we done for what is the question, part 1. Part 1.2. Yeah, so for these figures, we are going to generate a very similar figure, trying to compare the anchor regions versus the overall general background regions to see if they have different levels. To see if they have any difference in gene expression levels. Okay. One additional thing for part 2.3 specifically is it want to identify not only the intersections, but also if there's any motifs for these intersections. To detect those motifs, we are going to use this find motif genome. And this is something we installed from a software called Homer. So for this question specifically, we want you to load another conda environment because they have some version conflicts. So remember to, like I said in the beginning, remember to use this one for part 2.7 only, for 2.3 only. Sorry, I was saying Mandarin number 3. And then if you come moving on to part 2.4 and other parts of the homework, go back to the same conda environment. Try to keep that in mind. Okay. And then after everything is successfully finished, you would see your result files under motifs folder. And then you can just download all of them. You will see results for different chromosomes, for different motifs, different regions, and you will also see a summary web page. Try to look into this web page, make some screenshots, and explain what you see from that results. Okay. So that will be our part for, that'll be all for the part 2. And now the only thing left is the part 3. And I think now is a good time to stop recording. Let me see if there's anything I want to keep in mind. Give me a second, let me check my note. Yeah, I'm repeating myself, just trying to remember to switch between different locations. That's all you need to know. Oh, another thing I forgot to mention is that for when we talk about the sparse matrix in 2.1, we are saying, you know, now if the matrix is very sparse, it's a good idea to save the information in a sparse format. Why don't we always do that? It's because apparently if you have a lot of values, you know, instead of 5%, boxes has values, then imagine you have 90% of these are non-zero values, and you save the same matrix in this way. It's not that efficient anymore. It's not that efficient anymore. So you probably do not want that. And so you need to be clever on looking at your data. Some of the data is very sparse. You want to use this, some of them not. For example, here, if you look at this figure, even though you see here are mostly almost blank, you still have some values, right? So you imagine if you put this thing into a sparse format, that would actually increase the resource you need to do further calculations. So you probably, you know, one very frequent thing people do is to, you know, do some clipping. You know, we just filter out all these small values and we set them. We just force them to be zero, so that when you do sparse compressing, it wouldn't wouldn't take too much space for those information. Okay, this is just one thing I forgot to say. Okay, so that's all I have for part 2. And let's take a break and let's move on to the final part.